Hello everybody, my name is Sam Schmidt, a student in Dr. Woldering's lab at MSU. Today I'm going to be talking about coupling brain perfusion screens and next-generation sequencing to identify blood-brain barrier binding antibodies. Authors shown here. Why do we care about blood-brain barrier binding antibodies? They're useful as a therapeutic for many different neurological diseases. However, they're not always necessarily easy to find, mainly because in vitro results do not always accurately predict in vivo results, and when done in vivo, you get a large amount of background noise, and you have to use a random selection of clones to try and find the antibody you want. This potentially hides a lot of very useful clones. And another reason we're going to look at brain perfusion screening here is that intravenous application of phages in rats has a very short half-life of about 15 minutes. So brain perfusion screens take care of the in vitro result because brain perfusion screenings are in vivo. They also take care of intravenous application because they're not. Next generation sequencing helps alleviate this large background noise and random clone selection, hiding potentially useful clones. As a quick walkthrough, you take your phage library that you want to screen against, you perfuse it through the brain, you harvest it, you recover your phages, you send some off to be amplified, in this case CDRH3 amplification with their primer cocktail, and you sequence it with NGS and antibody mining software. And that allows you to get your clone selection, and you also amplify your pool. In this case, they run it 4 times, and you take a sample after each one to get a subset of your clones. So you can see that here with percentage of round 3 showing the clones here, round 4 just specifying that they used significantly less than an R4-12, perfused significantly smaller amount of phage. So here the 2 important phages I've highlighted as clone 4 and clone 40 because both of these clones bind the blood-brain barrier. This is shown down here in this micrograph. You have this ABN as a negative control, and you see almost no binding compared to the lectin, which should bind everything very well. OX26 is a positive control, and you see very good agreement between it and the lectin, as well as these 4 and 40 clones show a good agreement binding between where they're bright red and where the lectin is bright green. So that shows that these 2 clones are in fact binding the blood-brain barrier well. The authors also state that neither of these 2 clones were picked from their random selection, and these were a result of the NGS screening, and use this as an argument for why NGS is useful for finding blood-brain barrier binding antibodies in brain perfusion screens.